Hi, here we go. We are jumping in and we're going to talk about the process of science, what science is, how we gather information about the world, what happens when somebody's wrong in science. They claim something that is incorrect or later found to be false. So that's where we're going today. So science is a really broad field, but it's not just a body of knowledge. Well, most of what we're covering in our course and while you're in the educational world is learning about science that has already happened. It's also a process. Science is a process, right, of gathering new information about the natural world. And that can be things like gathering observations, describing phenomena, carrying out controlled experiments. There's many different avenues that science can take, but the key is that we are using empirical, evidence-based reasoning to describe the natural world. So what we learned about in class at one time somebody was asking questions about it, gathering data, and then made a conclusion based on their data. And then it ended up in our textbook. So the scientific method, while this sounds like a rigid process, you have to go from step 1 to step 2 to step 3, like following a recipe, it's not that inflexible. And using that term, scientific method, sometimes leads to people imagining or sometimes leads to people thinking that the scientific method can only be used if you're in a lab or to a very specific, narrow range of questions. But realistically, the scientific method is a very flexible process, and we can use this process to explore many different types of questions and thoughts and events and phenomena. So I like to think of this scientific method more of like a scientific process, or even more accurately, the process of thinking like a scientist. So kind of the key components of this are, first of all, making observations. And this can just be something as simple as, I wonder why I see all of those plants of that one type flowering at one time. Or I wonder why people who drink from this one specific well are tending to get sick. From your observation, you develop a hypothesis, an explanation for what you observed. Usually the hypothesis is grounded in what we know, current research that's published, right? It's just hypotheses are not an educated guess. Hypotheses are founded in prior knowledge that's been gathered about that topic. The hypothesis also leads to a prediction. So you have to make a prediction that informs your experiment. The key here is that without making a prediction about what your experiment will show, the results of your experiment, there's no way to really make any sort of determination about whether or not your proposed explanation of this observation was true or not. So we make a prediction based on what we already know, what's already been published. Then we conduct an experiment to test that prediction. The whole point of the experiment is to gather data, collect information that we can analyze using statistics, which helps us to draw conclusions from that data, to see patterns in the data, to see relationships in the data, to see differences between different groups in our experiment, which can then in itself lead to a new observation, or perhaps our conclusion doesn't support our hypothesis, in which case we could start this process over again. So once we get started, this process doesn't necessarily continue in this linear fashion from observation, hypothesis, prediction, experiment, conclusion. For example, one observation might lead to several hypotheses, and each hypothesis has a testable prediction and many different types of experiments that can be used to test that prediction. And often the conclusions, the information that we gather from that experiment, the conclusion that we draw from that data that we gather can be used to either make a new observation or refine our hypothesis, with the end result usually being increasingly precise conclusions about what we know. All right, so this is very hypothetical right now, theoretical if you would. Let's go through a very specific example. let's use the example of having a cold and echinacea. I kind of wanted to use a COVID example, but I thought that might hit a little close to home. So let's use this example as well instead. When we make observations, we're usually looking for, or we usually are noticing interesting patterns, maybe cause and effect relationships, and sometimes these observations can be anecdotal. So, for example, an anecdotal observation could be that we know many people that have claimed to observe that consuming the extracts of echinacea reduces the likelihood of catching a cold or reduces the duration of cold symptoms. So they feel better faster when they take echinacea. So that's our initial observation. And so, that's interesting. Let's explore this phenomena and see if we can understand what's happening with echinacea in the common cold. So let's say our hypothesis here is that echinacea reduces the likelihood of catching a cold and the duration of those cold symptoms. And if we were investigating this question, we would want to make sure that this hypothesis is grounded in prior research. So maybe we came across other publications that indicated that there could be a relationship between taking echinacea and the duration of cold or the likelihood of catching a cold. Okay, so we don't just pull this out of thin air, we base hypotheses based on what we already know about a system. Our hypothesis then helps us create a testable prediction. Oftentimes, these testable predictions come in this if-then format, but the idea is that it's to help us guide our knowledge in new situations. So we propose a situation that will give a particular outcome if our hypothesis is true, but will give a different outcome if our hypothesis is not true. So for example, the hypothesis that we had about echinacea and catching a cold and duration of cold, that leads us to two predictions. If our hypothesis is true, then the likelihood of catching a cold should be lower for individuals that take echinacea, and the duration of their symptoms should be shorter. And if our hypothesis is true, that means that individuals that don't take echinacea will have a higher likelihood of catching a cold. And if our hypothesis is true, individuals that don't take echinacea will have a overall longer duration of their cold compared to those that take echinacea. So now we have our predictions, what we would expect to see if our hypothesis was true. Now we can go and we can set up an experiment to test these predictions. Okay, so to test the hypothesis, echinacea reduces the likelihood of catching the common cold and the duration of symptoms. One study, this has been studied many times, but one study performed an experiment with about 437 people. These people all volunteered to be exposed to cold viruses. They all had cold viruses dripped into their noses. They consented to this. They then stayed in a hotel room for 5 days, and these people were examined by doctors and were checked for any cold symptoms. This is actually a fairly unusual experiment to expose human participants to an illness on purpose like this. If that is done on human participants, they were always informed of the risks and given informed consent. There's a decent history where that was not always the case. See Tuskegee syphilis study if you want more information on that. But be warned, it's pretty rough to learn about that, but it's good history to know. So once we have formulated a hypothesis that generates a testable prediction, we then conduct a critical experiment. And the point of this experiment, again, is to make a determination about whether our hypothesis is supported or not, based on the data that we collect. But the whole point of this experiment is to make observations that either allow us to say our hypothesis is supported or a hypothesis is not supported. Experiments can happen in many different settings. It can happen in the lab, running samples in a very controlled environment. Experiments can occur out in the field just by making observations. Or you can take your experiment out of the lab into the field. For example, this is me running my experiment from graduate school. This was one of my projects looking at the effect of coloration on predator behavior. Okay, so what are the critical components, or what are the crucial components of an experiment? So this is just a little vocab I want to go through with you guys real quick. In order for an experiment to be considered valid and to be powerful, meaning it's robust enough to give us good information about this phenomena, good experiments have at least these 4 components. So first being a treatment, meaning that any experimental condition that is applied to research subjects. Dosage of echinacea given to somebody in our example of echinacea, right? How many milligrams of echinacea did they get a day? Could be a treatment. And then the experimental group are subjects that are exposed to a particular treatment. So for example, in our echinacea scenario, those individuals that were given echinacea would have been part of the experimental group. Other individuals, though, may have been given a placebo. So a placebo, or sometimes called the placebo group, is the other portion of the subjects who did not receive echinacea. We want to minimize the differences between our groups, though. So if they're not receiving echinacea, sometimes just the act of taking a pill can have an impact on our physiology. And so we give them a placebo, basically a fake pill, there's nothing in it, in order to have them still go through the act of taking a pill. Okay, so those individuals that take the placebo, we call them a control group. So these are individuals or part of our experiment who are treated identically to the experimental group, but they don't get the actual treatment, in this case the echinacea. Okay, so placebo means they aren't getting the echinacea. The experimental group are getting echinacea. And then variables are any sort of characteristic in your experiment, in your system, that might change. Sometimes we have variables that we want to change. So independent variables are variables that can be observed and measured and whose value can be changed as required. So for example, an independent variable could be the dosage of echinacea somebody is given. Maybe they're given 10 milligrams versus 20 milligrams versus 30 milligrams. A dependent variable is a variable that we measure in response to the independent variable, to changing the independent variable. So in our echinacea example, the dependent variable could be how long the symptoms of the cold last. Other variables, though, that typically experimenters try to minimize the difference between their groups, right? So let's say, for example, we had only young, healthy people in their 20s in the experimental group, but we had immunocompromised people in our control group. That's a variable that would not be equally distributed between these two groups that could impact the outcome of the study. So we want to minimize any sort of differences between the experimental group and the control group to make sure that any sort of change in the dependent variable that we find is due to the independent variable and not to a variable out of our control, like being immunocompromised versus having a normally functioning immune system. Okay, so let's talk about this echinacea study. So here's our treatment. We had individuals randomly divided into 4 groups. Okay, so that's key, random. This helps to minimize any sort of difference between the groups that might exist as part of just maybe their background, where they come from, their health, their diet, things like that. 2 groups took a pill daily. So these 2 groups here took pills before exposure to the virus. Groups 3 and 4 did not take pills before exposure to the virus. So this only group 1 took echinacea for a week prior to exposure to the virus. They were then exposed to the cold virus, and then they continued to take echinacea tablet daily. Group 2 took a placebo for a week before the virus. They were then exposed to the cold virus, and then they continued to take a placebo for a week. Group 3 and Group 4 did not take any pills, either echinacea or placebo, before exposure to the virus. They were then exposed to cold viruses at the same time. Group 3 took echinacea tablet only after being exposed to the cold virus, and then Group 4 only took a placebo after being exposed to the cold virus. And what's key to this study is that this is called a double-blind study, meaning that the individuals involved in carrying out this research, the doctors, and the participants themselves did not know what group they were in. They did not know if they were taking a placebo or the echinacea. The doctors nor the participants knew which group they were part of. It's usually good practice to conduct a double-blind study because we can accidentally, subconsciously send information to another person about their health or their well-being and our expectations for their symptoms when we know if they're part of one group or another. So let's see what they found out.